DNA's scientific term, deoxyribonucleic acid, is the genetic material of cellular organisms and most viruses, the genetic molecule which is used to encode genetic information for all life on Earth. In 1953, a young American geneticist named James Watson and his British colleague Francis Crick had a brilliant insight into the building plan of the DNA molecule that established several things. It demonstrated that DNA almost certainly contained the hereditary material of cells and organisms. It revealed how cells could copy DNA to pass it along to their offspring, and it showed how the molecule might change through mutations, which makes evolution possible. The new structure proposed by Watson and Crick caused a sensation in the scientific community because it made sense of some of the physical measurements that their British colleague Rosalind Franklin had made of DNA. DNA is a long chain of polymers, a small compound called nucleotides. Nucleotides are composed of a base, sugar, and a phosphate group. The phosphate joins the sugars in a DNA chain through their hydroxyl, the group by phosphodiester bonds. There are 4 different types of nucleotides found in DNA, differing only in the nitrogenous base. A is for adenine, G is for guanine, C is for cytosine, and T is for thymine. The bases pair in a specific way, A with T and G with C. The bases are classified based on their chemical structures into 2 groups. Adenine and guanine are a double ring structure called purine. Thymine and cytosine are single ring structured termed thymidine. The studies that measured amounts of the 4 DNA building blocks, nucleotides in different species, launched a new era in biology. That shifted the focus toward the interaction between different types of molecules in cells. Within cells, DNA is organized into structures called chromosomes. Eukaryotic organisms, for example, animals, plants, fungi, and protists, which store their DNA inside the cell nucleus, while in prokaryotes, for example, bacteria and archaea, is found in cells' cytoplasm. In human cells, DNA is contained in the nucleus, arranged in 23 pairs of chromosomes. The 23rd pair of chromosomes determines the sex of the individual and is composed of either 2 X chromosomes for a female or X or Y chromosomes for a male. Since most eukaryotes are multicellular organisms with many cell types, gene expression must be controlled so different cell lineages develop differently and remain different. A brain cell is quite different from a liver cell because it contains different proteins, even though the DNA in the 2 cell types are identical. During development and differentiation, different sets of genes are switched on and off. Hemoglobin, for example, is only expressed in developing red blood cells, even though the globin genes are present in all types of the cell. Genetic engineering technology has made the isolation and manipulation of eukaryotic genes possible. This has given us some insight into the extraordinarily complex process that regulates transcription of eukaryotic genes and allowing fertilized egg to develop into a multicellular, multi-tissue adult. Basic eukaryotic cells contain the following. 1. Plasma membrane. 2. Glycocalyx. 3. Cytoplasm. 4. Cytoskeleton. 5. Presence of characteristic membrane enclosed subcellular organelles. Characteristic biomembranes and organelles. Plasma membrane. Nucleus. Mitochondria. Chloroplasts. Rough endoplasmic reticulum. Ribosomes. Smooth endoplasmic reticulum. Golgi apparatus. Lysosomes. Vacuoles. Peroxisomes or microbodies.